Perfect.
presentation.
Okay, great. And for the invite, of course, I hope everyone's enjoying the conference so far. So my name is Hajer. I'm based at Imperial College London. I'm from Tunisia and Morocco, so both descents. And the topic of my talk today is going to be around multimodal data in healthcare and clinical research, more specifically, like what benefits do we see in it and the challenges that we encounter. All right, so a little bit about me, my background. I got my bachelor in computer science at Drexel University in the US back in 2018. So that's where I majored in comp sci with a concentration in AI security, and I did a minor in neuroscience as well. And I did 3 internships, like 6 months, and each of them. So it was like mostly like web and mobile app development, and the third one was about like in IT network security. And then I went on to Ernst & Young in the New York City headquarters, and I was a full-time cybersecurity consultant before then moving to London and doing my master's of research in experimental neuroscience at Imperial College, where I still work there at the moment as a researcher and a data manager. I'm also a really big advocate for volunteering and mentoring. So I'm a mentor at Imperial, as well as a mentorship program lead at AfriCode, which is a nonprofit organization that really looks after students from Africa and all over the world. So I'll start by talking a bit about projects that I've done in the past or ones that I'm working on at the moment. So as I mentioned earlier, my master's was a master's of research, which means that you get to work on a few projects throughout the year, which I was very lucky to do. So the first one, I wanted to look into the effects of putting someone in a virtual reality experience and how does that affect their pain perception. I'm happy to talk about that project later if anyone has any questions. We also, apart from the VR experience like environment, we also gave them an auditory stimulation at a certain frequency that is known for the brain to settle on that specific frequency when there's top-down modulation of pain. The second project, this is sort of like the second and third project. So we used MRI data, so magnetic resonance imaging. So that's when you put someone in the scanner, we scanned their brain while they were doing a specific task. And we wanted to use a support vector machine to see if we can differentiate between the brain connectivity of the patients and the healthy people, if the states are discriminable between these two different groups, and if we can tell based on their brain connectivity which tasks they were doing in the scanner. So they were asked to do 2 different tasks. One involves mental planning and one involves just counting a few items on the screen. And then this third project that I'm currently trying to work on, we want to see if we can use Bayesian statistics to sort of cluster and subtype and stage anxious and depressive profiles using just questionnaire data. So people are presented with questionnaires of anxiety and depression. They're known as the GAD and PHQ. And based on their answers, we treated data cross-sectionally, of course, and we tried to see in an unsupervised way if there's any way we can identify subgroups of of people who are depressed and anxious, and try to model basically which question starts like telling you about the progression of this neuropsychological condition. And then this last one, so I mentioned I was a data manager, and so I am responsible for the front and back end of the database of the tissue bank at Imperial. So the tissue bank is where all the brains get donated after a person dies. So the patients that we usually donate their brains to us had multiple sclerosis and Parkinson's during their life. And also, of course, we get people who are healthy donating their brain. So, of course, if you have to compare to what a healthy brain looks like. So I'm responsible for that. So it's a lot of, ASP.NET and SQL and all that stuff. So I talked about a lot of projects so far, and there's only some of the things that I got to work on. And as you probably noticed, these projects use different types of data within clinical research. So for instance, we have PROs, patient reported outcomes. So again, these are something like questionnaires, so they can measure like disability scores, anxiety levels, depression levels, all that. We have omics. So this field or data modality refers to everything that we look at. Like basically you get to analyze different types of data that have to do with like, metabolites and different types of proteins and molecules, depending what you want to look at, of course, genomics and all that. We have PET, DTI, and MRI. So PET is positron emission tomography. So that's when you inject a ligand in someone's body, and then you want to monitor the uptake in the brain or in the body of a specific molecule. So glucose is one of them, for instance. DTI and MRI mostly look at the brain structure or how the different hubs in the brain talk to each other while the person is either performing a task or is just at resting state, not performing anything in the scanner. We have eye imaging, which is becoming more and more famous recently, especially in multiple sclerosis. They're finding that people with MS have certain like cell death at the back of their eye. So that's something that people are investigating right now, which I think is quite interesting. And we have real world and data and wearable data. So this is really, it helps us like learn more about the patient experience, especially when you're dealing with sensors. So it could be collected, for instance, in a living lab. So this is, a lab that's been built specifically for a person to live in just for like, 2-3 days, just to monitor their activity. It's full of sensors everywhere. So they put sensors in the bed or around the room to sort of analyze the movement of the patient or measure stuff like sleep disturbance or infection diagnosis in the home. And so it's really good because you can like derive key, for instance, like dementia-related measures that, for instance, the UKDRI is dealing with right now. So again, lots of data, but the thing that you really want to think about is, how do I approach this? How do I analyze this? You can choose multiple analysis approaches to project. So I'll just talk about a few here, the main ones that people use. So we have frequentist statistics. So in this framework that most of the scientists and researchers have been using, in this framework, we don't really assign a probability to the hypothesis. And there are no priors to be set compared to the Bayesian that I talked about a little bit earlier, and then I will also explain now. So you pose a null hypothesis and you either accept it or reject it, okay? And so for the Bayesian one, here we try to answer the question in a different way. So we say, what is the probability of the hypothesis given the measured data? So basically it's sort of like data driven, and it keeps getting updated the more data you get. So it's not as biased and it's quite robust. So again, like here, it's going to use the probability of the data and both hypotheses that you're trying to test. And you can also use the probability to represent the uncertainty in any event or hypothesis. So when you're using Bayesian here, you're sort of forced as a scientist to confront your assumptions because you need to set your priors, of course, and interrogate these assumptions and justify why you chose them. And then we have machine learning and AI. So again, this has been more and more of the hot topic of the past 10-15 years. So an important question to consider here is knowing from what angle are you trying to solve your problem. So in clinical research, are you doing this? Are you using ML and AI to sort of as a prognosis purpose or diagnosis? Are you trying to understand more about how a certain biological process works? And so how also will you have clinicians trust the output that you have? And this would really dictate how you as a data scientist build your model and use the data and the results that you get. So again, there are more and more claims that Bayesian stats are a lot more robust, especially in clinical research for a few reasons. One of them is the fact that it can handle data missingness quite well. And data missingness is a big thing in healthcare. As you know, data collected is not collected in the best way. Sometimes it differs between sites. And so the fact that it can handle data missingness is because it is able to estimate the posterior distribution of a missing data point. All right. So, and also it's fundamental for ML and AI. So it ties in together quite nicely. So then I wanted to just walk you through a few projects that successfully combine multimodal data. So this one, this paper, Jacobs et al. And Cole et al., they basically combined two different tools. So they explored the links between an MRI-derived brain age, which I will explain in a little bit, and blood-derived cellular senescence, which is biological aging, okay? So let's start with brain age. So the authors in this paper, the Cole et al. Paper, they use machine learning to predict the patient's age based on the brain structure, different feature they extracted from MRI scans. And then they subtracted the chronological age, the actual age of the patient, from the brain predicted age based on the morphology of their brain, okay? And so the higher the difference, the poorer the brain health is. And so they found that people who had multiple sclerosis had older appearing brains, okay? So it means that the predicted age was a lot higher than the chronological age. And so, which means that, it was quite sensitive to disease progression and disability that is caused by MS. And they wanted to correlate that with cellular data that they collected. Specifically, they were targeting T cells. And so they concluded that these T cells were a lot more frequent in older appearing brains that were rated by brain age. And then they were also associated with worse disability scores. So this is a PRO, okay? So the EDSS score is a score that assesses the disability of a patient that has MS, which suggests the roles of T cells in the disease progression. So, again, like we put a lot of types of data together and we tried to come up with sort of a model to understand the disease better. So in this paper, the authors combined MRI and PET and PRO. So they explored relationships between pathology-related metabolites using spectroscopy or MRI, and then cells associated with neuroinflammatory response using, again, PET. So you inject a ligand that is taking up those specific activated cells that you wanted to look at and monitor. And they correlated the findings with disability scores. So these are the PROs. And so again, they found that the cell activation that is measured by PET was associated with a very high concentration of the pathology-related metabolites that they were monitoring using the MRI and higher disability scores, okay? So this was more of a future direction in my first project that I mentioned, the virtual reality effects on pain perception. And so, as I mentioned earlier, apart from the VR experience, people were hooked to a headphone that was playing a track at a frequency of 10 hertz. So the idea was to measure the participant's brain wave during this pain relief process or brain modulation. And the auditory stimulation that they're hooked on at alpha frequency, so alpha is about 10 hertz, has been found to reduce this experimental induced pain. So this is, again, like, future direction, combine how they score the pain with seeing if their brainwave is actually changing due to the stimulation. And then in this project, which is actually currently being designed by a lab that I'm working with right now, so we're trying to do like a whole brain imaging. So you image a whole dead brain using ultra high field MRI. And it would really help you capture a lot more detail than like a normal, like if someone was alive, basically, because when you put, just the brain in the scanner, you have a lot more time to scan it for. And so you get a lot more detail as opposed to, if the person's alive, you can only keep them in there for 2 hours. And so we do a whole brain imaging, so again, using MRI, and then we take him down to the cellular level to drive the omics, so genomics, proteomics, transcriptomics, and metabolomics, so all of these measures. And the aim is to map all this data together and find new pathogenic mechanisms that would help with patient stratification. And again, it would help you with better drug repurposing and really tell you, like, how can we leverage MRI imaging to then reflect pathological patterns at very early stages of the disease. So this is a really nice like workflow graph that I really like. It's showing you just a multimodal way of analyzing the data, investigating MS. So again, like we start with the brain imaging, like the MRI, and then we take it down to the staining and dissection level. That we derive the neuropathology profile, and then derive all the other measures. So as data scientists, you probably are aware of a number of issues that can be encountered when analyzing the data, right? So that includes data missingness and insufficient data disaggregation. So basically the data is not broken down by categories, inconsistencies in how the data is collected. So I wanted to focus on that today because I think it's quite crucial for us as data scientists and researchers to be aware of all the things that could go wrong. So we're better prepared to mitigate the effects, especially in healthcare, that the impact is quite high. And it's like people's lives on the line. So when either collecting the data or analyzing it. I'm just gonna spin through here because I will just explain it in a bit. So here, one of the first challenges is patient variability. So for instance, in the VR project that I mentioned, we have a few participants that played Muay Thai, which is a martial art. And so they were exposed to quite a lot of pain during, their daily life. Like, if they train like every other day, they would perceive pain as a lot less bad than normal people who wouldn't necessarily play that sport. And so they were asked to like rate the pain from 1 to 100, and they were rated as a 10 or like as a 5, which was completely different from how another person would view it. So it's completely subjective. So this patient variability is quite important to account for when you're analyzing the data. Something that we tend to do as researchers is just like, get rid of the outliers. But that could be a problem because when you get rid of outliers, you might be overlooking crucial, like, biomarkers or something that's like specific to like these types of patients, and just you're just not digging deep enough to see if there are like anything else that you would want to give your attention to. And so the solution is to make sure that your model is generalizable enough, but also that it encapsulates the variability that you encounter. Another challenge is fragmented and decentralized data. So for a lot of you who are dealing with, dealing data in healthcare, especially with like non-private. So basically, if it's the public sector, it's the data collected tend to not be, quite well centralized, stuff everywhere. The foundation of like making sure that your data is in one place is, it's not really there basically. So me as a data manager, I try to basically think of it as from a researcher perspective. So if I were to do research using this data in 5 years time, how do I want the data to be structured? And so there's a lot of issues now. It's stuff like how the data is linked, which is known as data linkage, how it's recorded, to make sure that it's in a fit format for research later. I, as a data manager, try to design the database and the front end in a way that I don't give too much freedom to the user, when they enter the information. So for instance, like stuff like stupid things like free text of entering what's it called, like drugs data, right? So do you want to see enter what kind of drugs the patient was on? Some people enter like different names for the same drug, like typos, stuff like that. And it becomes really difficult. Like when you scale it up, it becomes really difficult to analyze later on. And so it's stuff like that. You just like minimize the freedom, but also too much specificity can also harm how generalizable it is to get, different types of data. So standardization and interoperability would be a good solution to keep in mind when you're designing these kind of systems. A big thing as well in research is how different labs tend to analyze data in a different way. So you can basically have access to the same exact data, but end up with different results or not be able to replicate and reproduce the results that another lab did. So that's quite an issue. And so really the solution for this is make sure that open access code and open access research is one of your top priorities. So have a standardized exploratory data analysis, and you always want to ensure replicability by, for instance, like publishing your code and making sure that another person, if they were to read the paper, they would be absolutely capable of replicating the same results. And so this is something that people have been like looking into it more and more recently, because AI and ML and deep learning, it's been used in pretty much all fields, and in healthcare specifically, if you're trying to understand a certain biological process or do prognosis or diagnosis tools, then you really need to understand how you get the result that you're getting and be able to interpret it in the right way. So especially in diagnostic and prognostic tools. So does it make sense biologically when you get a certain result? So for instance, this is one of the outputs that I get from trying to model a certain, like the depression and the anxious profile. So here it's showing you like different biomarkers, which are, in our case, since we're using questionnaires, is different questions. And it tells you which question starts telling you a little bit like early on about the disease progression. So here the question, like the thing I want to ask is, does it make sense for question 0 to not become abnormal until the very end, like stage 13 of this neuropsychological disorder? So it's something that always, like, we shouldn't take the AI result just for granted and just like, just trust it 100%, because we also have the expertise as clinicians, and I'll make sure that we talk to them to ensure that we're communicating the right stuff. So explainable AI is the solution for this. You're making sure that you're understanding how your system works and how it's generating the results that it's given you, and constant communication between data scientists and clinicians. I saw someone in the chat actually ask something about how AI modeling helped or did not help COVID, catching COVID or diagnosing it recently in the past few years. So the MIT Technology Review published quite a nice article about that, looking about like different tools that either helped or didn't help just during COVID. The Alan Turing Institute also had a really nice article about this, and basically just wanted to take a snippet here to show you that AI is not always a solution. Sometimes it's a problem with the algorithm, sometimes it's the data. So for instance here, this tool was fed more scans, like pediatric scans, so like of kids, and so it was undesirably trained to learn to identify kids as opposed to if a person had COVID or not. So that's a problem with the data, obviously. The second example was all the scans were of people lying down, so it learned to identify the position of the person as opposed to if they had a serious COVID risk. And then this other one is having data from different hospitals being fed to the same system. It just learned really to identify which hospital do the scan come from based on like the font around the scan. And so these are like tiny, they're not tiny things, but like these are tools that we need to also like always take with a grain of salt and make sure that we're feeding it the right data and interpreting the result in the right way. And so just to summarize what I was talking about right now, but like making sure the communication is right between data scientists and clinicians. And so if there's no communication between them, neither of them are going to trust the other, right? We want to make sure that there's communication on both ends. So they're both like giving feedback to each other throughout this process. So I would call it like a stepwise collaboration. And so in that way, the data scientists would understand the implications of the choices that they make when they're analyzing the data. And then the researchers would make sure they would understand how to best leverage the potential of AI, because a lot of them are skeptics, and fairly so. The last challenge that I want to bring up here are the ethics. So are we interpreting AI, machine learning, and deep learning results in an ethical manner? So what I mean by this, I touched on this recently earlier. So when you're, for instance, telling a patient, you're at a higher risk of getting dementia or MS or whatever disease it is, what are you actually doing with that information? Are you just giving them that information without putting a measure in place to, for instance, like a vaccine trial or all that, or the ways of telling them how to mitigate or slow down the disease progression? So you can't just be giving them basically like a death sentence of like, this is how your life is going to look in like 5 years, without making sure that you're actually doing something about it. And so the first thing always is to make sure design ethics run the project before launching it. You want to assess the accessibility of it. So for instance, with like when you're doing like wearable data tools and technology, you want to make sure that however you're collecting the data, make sure that it's collectible on devices that are already out there, as opposed to, for instance, making your own device from scratch. So it makes it really less accessible for people. You assess the fairness and bias in your research, so you want to ensure that you touch on most ethnicities and populations, and not just like a certain like subgroup of people. Including and empowering patients, this is really important. So this goes under like PPI, so public patient involvement. So this is basically getting the patients themselves that are the most concerned with their research, really, to engage with you in designing the ethics, the objectives of your project, what they think is important, especially if you're doing something that is supposed to make their life better. And communicate the limitations. So again, if you communicate to someone, you're at risk of getting X, Y, Z in n number of years, then you really need to also tell them about the limitations of your like research and your tool, to because it's obviously like not 100% correct, right? So that's something that you really want to communicate always. Just a few acknowledgements here. Professor Adam Hampshire, Dr. Amy Jolly, and Richard and Roberta have been tremendously helpful the past few years. I learned so much. I came from a computer science background, and now I'm on like neuro and computer science, which has been a journey, a good one. And yeah, thank you for your question, for your attention. And please let me know if you have any questions.